Welcome to Chapter 1 of Pharmaceutical Chemistry. In this session, we will explore the scope, errors, and impurities related to pharmaceutical substances. First, we'll begin with a short introduction to pharmaceutical chemistry, what it is, and why it is essential in the field of pharmacy. Next, we'll move on to the scope and objectives of this subject, focusing on its role in drug development, safety, and quality assurance. Then, we'll study the types and sources of errors that may occur during pharmaceutical analysis. This includes systematic errors, random errors, and gross errors. After that, we'll cover the core concepts of accuracy, precision, and significant figures, all of which are essential in achieving correct and reliable results in lab calculations. We'll also discuss impurities in drugs, learning where they come from and how they can affect drug quality, safety, and efficacy. Finally, we'll spend time on the most critical part of this chapter, limit tests. We will understand their importance, basic principles, and the detailed procedures for the tests of chlorides, sulfates, iron, heavy metals, and arsenic. Subscribe to D Pharma Pathshala for more simple and exam-focused pharmacy lessons. Tap the bell icon so you never miss an update. Let's begin our journey into pharmaceutical chemistry, a subject that plays a vital role in ensuring the safety and effectiveness of the medicines we use every day. Pharmaceutical chemistry is a special field of chemistry that combines organic chemistry, biology, and pharmacology. Its main goal is to discover, develop, and analyze drugs that help treat diseases. It includes everything from how a drug is discovered, how it's absorbed, how it works in the body, and how we can make sure it is safe and effective for patients. Now let's talk about its scope. This field gives opportunities in quality control and quality assurance, QA and QC, making sure every drug that reaches a patient is pure, effective, and safe. Drug discovery, creating new medicines that can treat diseases more effectively. Industry, working in pharma companies that manufacture and test medicines. Teaching, becoming a lecturer or teacher in pharmacy colleges. Let's now understand the concept of errors in pharmaceutical chemistry. An error is a deformity or deviation in any measurement that occurs due to internal or external factors like faulty instruments, human mistakes, or improper methods. In pharmaceutical analysis, even a small error can lead to wrong dosage or unsafe results, so it's very important to recognize and control them. Errors affect the accuracy and precision of our analytical results. Sometimes the result may be close to the true value, accurate, or it may be repeatable but not necessarily correct, precise. Now, errors are divided into two main types. The absolute error is simply the difference between the measured mean value and the true value. It tells us how far off we are. The relative error shows how significant the absolute error is compared to the true value. It's usually shown as a percentage or parts per thousand. We also consider significant figures. While reporting measurements, they reflect how accurate and reliable a number is. Understanding these basics helps us make better, more consistent pharmaceutical measurements and decisions. Let's begin with the concept of accuracy. Accuracy means how close your result is to the actual or true value. In science, since the exact true value is hard to determine, we often use an accepted average from multiple labs. The lower the error in your result, the higher the accuracy. Moving on to precision. This is about how close your repeated results are to each other, regardless of how close they are to the actual value. A measurement can be very consistent or precise, even if it's not accurate. For example, if you measure water content in milk and get values like 88.3, 85.4, and 87.9, they are grouped closely and show precision. Finally, significant figures are used to express your result in a way that reflects its true precision. They include all digits that are known with certainty plus one estimated digit. For instance, the number 8.73 has 3 significant figures. This practice helps maintain accuracy and reliability in scientific reporting. To reduce or minimize errors during pharmaceutical analysis, we must take several precautions. First, always begin by calibrating instruments. Even the best equipment can give wrong results if it isn't calibrated regularly. Second, ensure trained and skilled personnel are handling experiments. Proper training reduces human mistakes like incorrect readings or improper handling of reagents. Third, always select the right materials, like suitable glassware and high-quality reagents. This helps avoid compatibility issues and inaccurate results. Fourth, focus on eliminating contamination. Clean apparatus and pure chemicals ensure that impurities don't interfere with the results. Fifth, a clear understanding of chemical principles and reactions improves how we interpret results, minimizing misjudgments. Finally, following a proper standardized method is crucial. Deviating from standard procedures often leads to inconsistent or wrong results. By applying all these practices together, we can achieve more accurate, precise, and reliable outcomes in pharmaceutical chemistry. Let's understand what impurities are in pharmaceuticals. Impurities are unwanted or unexpected substances that enter the product during any stage of manufacturing, handling, or storage. These impurities can change the effectiveness, safety, or quality of the drug. In an ideal world, we want drugs to be 100% pure, but in reality, it's very difficult to achieve absolute purity. Small traces of other substances may get mixed due to production methods, incomplete purification, or chemical reactions. These impurities may have toxic effects or reduce how well the drug works. So it's a big challenge for pharmaceutical companies to remove or control them. To handle this, pharmacopoeias, like the Indian Pharmacopoeia or USP, have set specific limits for how much impurity is acceptable in each drug. By following good manufacturing practices and regularly testing for impurities, we ensure the medicines we take are safe and effective. Impurities in pharmaceuticals can be introduced at any stage, from manufacturing to storage. Let's look at these stages one by one. First, during manufacturing, raw materials may carry contaminants like heavy metals or residual salts. The reagents and solvents used must be of high purity or they risk adding undesired substances. Even the equipment used like metal tanks or glass-lined vessels can react and leach trace metals like lead or bismuth. Sometimes intermediate compounds formed during reactions stay behind as impurities if not completely removed. Plus, the manufacturing environment, especially in industrial zones, can contribute airborne dust or gases. Second, during purification and processing, the same risks apply. Even the chemicals used to remove other impurities like adding BaCl2 to remove sulfate can become a new source of contamination. Solvents, especially water, must be clean and tested. Equipment like filters and dryers must be properly maintained to avoid adding rust or moisture into the product. Lastly, impurities can enter during storage and packaging. Poor quality packaging materials can leach chemicals into the product, and improper packaging processes may leave products exposed to air, moisture, or microbial contamination. This is especially serious in sterile products like injections or eye drops, where microbial growth poses health risks. To maintain drug quality and safety, strict control and monitoring of all these stages is essential. Let's now understand the effects of impurities in pharmaceuticals and why they are a major concern. First, even though some impurities may seem harmless, if they cross a certain limit, they can reduce the drug's potency. This means the medicine may not work as intended. Second, some impurities cause incompatibility by reacting with the drug substance, leading to chemical changes or deterioration of the final product. Third, certain impurities can interfere in chemical reactions of the drug, changing its behavior and reducing its effectiveness or stability. Fourth, even small amounts of toxic impurities can accumulate in the body over time, causing harmful effects later. This is known as cumulative toxicity. Fifth, some impurities create a favorable environment for microbes, leading to bacterial or fungal growth. This not only spoils the product but also makes it unsafe for use. Sixth, some impurities act like catalysts. They speed up the degradation of the drug, reducing its shelf life and effectiveness. And finally, unstable impurities like those with hygroscopic or oxidizable nature, can change the physical appearance, taste, smell, or even texture of the drug. This makes formulation and patient use difficult or unpleasant. That's why strict impurity control is a must in pharmaceutical manufacturing.